So talk to us a little bit about other TRK-Fusion approaches to cancers, maybe a little bit about LOXA195 and TPX0005.
Larotrectinib and entrectinib are not the only agents that are targeting NTRK. There are a number of additional agents that are being developed. LOXA195 is one example. Was just reported at AACR in March and April of 2019. And this too works on TRK fusions. Response rates in those with an established TRK kinase abnormalities, close to 50%, 9 patients out of 20. Again, a variety of different cancers. My initial read on the toxicity is that it may be somewhat harder to deal with. Nausea and vomiting, myalgias, abdominal pain, in addition to fatigue and edema and constipation. So that always is tough when we're dealing with an oral agent that needs to be given continuously on a daily basis. It's unclear in patients with previous exposure to entrectinib or larotrectinib how well these newer agents are bound to work. At least for LOXA195. That's not the case with repotrectinib. It's another new agent. Again, something of a dirty kinase, works on multiple different targets. Response rates to date are 80%, quite a bit higher, 66% in those with prior exposure to other TKIs, including crizotinib. It works on ROS1 and specifically works on the mutation, resistance mutation, G2032R, which is probably one of the main mechanisms of resistance in ROS1. And then there are a number of other agents that have actually been used in other malignancies like midostaurin, FLT3 acute leukemia, and regorafenib in colorectal cancer, the latter starting out as an angiogenesis inhibitor. These two appear to have some activity in NTRK, but it's completely unclear so far just how active they will prove in this patient population. We're really just awaiting the phase 1 and phase 2 data. A new era has dawned. When I started out as a medical oncologist in the late 80s, it's over 30 years ago now, I couldn't have dreamt that we would have targeted therapy of this sort. And now, for the first time, we've witnessed the approval, marker-specific approval of a new agent, larotrectinib, that's histology and tumor agnostic, and clearly is pointing the wave to the future, the way I think we're going to practice increasingly. And there are a number of trials that are ongoing. They're actually addressing this. The MATCH trial through Eastern Cooperative Oncology Group and jointly with the other cooperative groups is looking explicitly at marker-specific therapy, establishing the role of various agents in a particular tumor marker and allowing individuals with relatively rare malignancies or different malignancies to enroll on a single study that essentially examines 15, 18, 20 different markers and 15 to 20 different agents. You could never, no single institution could ever mount studies of this sort. The patients are just too rare. But if you do it in the context of a MATCH trial that looks at multiple different markers and then matches up the patient with the appropriate markers for the appropriate targeted therapy, it makes sense. And it's really a brilliant academic, community, industry, and federal partnership that's come through that I think can address the needs of many of our cancer patients for whom standard therapy, at least traditionally, does not exist. Larotrectinib's approved. I anticipate entrectinib's approval sometime in the summer of 2019. And then there are other agents such as repotrectinib and LOXA195 that will probably be approved in the next year or two based on their activity, both of which have shown responses in individuals who've had prior NTRK inhibitor exposure. And there may be a role for other agents that are already established in other malignancies, such as regorafenib and midostaurin.
You know, as all small molecule inhibitors, we do expect that there will be resistance. And again, as the experience to date has shown, you know, these patients stay on for an awfully long time. but some of them do develop resistance, and when we biopsy these patients, we've shown that these patients develop what are called solvent front mutations at the site of the ATP pocket. They also develop what are called gatekeeper mutations. There are other alterations and other mutations that are less common, and we do think that there may be kind of alternative pathway signaling processes that may be leading to resistance. But the most common mechanism of resistance is solvent front.
And my understanding is these were expected at the time
These
of
were
the design.
Of the design. We were expecting this, and kind of another first in this approval was that we began the phase 1 even before we got the approval of Lero.
The phase 1 of which drug?
Of LOXO-195. And began enrolling patients who were becoming resistant. In fact, Alex and the MSK group actually published a small case series of patients who developed mechanisms of resistance, and were able to, through kind of a compassion IND, be able to put those on before we even started the phase 1 to actually see responses in these patients.
So were those resistant mutations just for the 195 study just coming from the larotrectinib trials,
No,
or were they also
no.
coming from the entrectinib?
So they were also from patients who also received entrectinib. So to date, I mean, I can't share with you all the data, but Dave Hyman did present some of the preliminary data that we have at AACR showing, significant responses in a number of these different subsets of tumors.
So if you could, I have a question for you about that. Is when you have these resistance mutations and you have a patient who has it, are you seeing progression of multiple sites, or is it usually just like one site starts to grow while the other ones are still responding?
Yeah.
Just so that people who are starting to get this drug know what they're looking for.
Yeah, that's a really good question.
I mean, I know they're not that many.
In my experience, and we haven't, Dave Hyman's probably put the most number of patients on the resistant population. In my experience, it's variable. There are patients who seem to be just progressing everywhere, and there are patients we were following the target lesion, right?
Right.
And target lesions, and overall they progress by RECIST. And when we re-biopsy those lesions that seem to be progressing, they had the solvent front, but there were other diseases. And as you can see,
that's really interesting, because many times in other kinase inhibitors, many for us, when we get resistance, many times everything's responding, and there's just one area that seems to have figured it out.
So again, I think it's variable, and we haven't looked at the full data set. In my experience, it's a handful of patients had just one area of kind of progression because of how we follow these patients by RECIST criteria. There are some other patients who seem to, it was less definitive because they had lung mets. They may have small lesions, and all those lesions seem to be growing on top of their target lesions and so forth.
And what do you know about, say, the toxicity of 195 compared to Lero?
I think that's just preliminarily right now. The profile is very similar to LOXO as the larotrectinib trial.
Okay, great. And so what about TPX-0005? Can you tell us a little bit about that? Because I know that's slightly different because it also inhibits ROS1 and ALK. So it's
correct.
Like
it's similar to entrectinib. There's very little data on its effects on NTRK. Clearly, I think that some of the data was presented on ROS, and the efficacy seems very good. 80% response rate, my understanding.
Wow, great.
So.
And can you talk about other kinase inhibitors that are out there that also have been documented to inhibit TRK, including things like cabozantinib, regorafenib, crizotinib? They're more general inhibitors. I know that they target many things, but can you tell us a little bit about the experience with TRK in those cases?
So there's very little experience. There's very little been reported on these other agents, and from what our understanding is that they're not really true TRK inhibitors, and they're kind of IC50s relative to entrectinib and larotrectinib are very different. And so my recommendation would not be to if you find an TRK-Fusion patient, give them crizotinib. You know,
Right.
there's an approved drug now for TRK-Fusions, which is
So neat, right?
Yeah.